The insistence on diversity, ecological theory suggests that diversity, plant diversity, will result in sustainability with a stable system over time, and it will also result in a resilient system over time. How to prove that? They've done it ecologically in grasslands, but how to prove it in an agricultural ecosystem is far more difficult. So we set out to measure, and it's one of the discussions about soil ecology is how do you measure it, what's good and what's bad. So we set out, and if I can remember them all, first was actually measuring production, which we did with a half square meter, harvested the green material, and at the end of that first year planting here, we had, again, it's a hectare equivalent. In our fourth diverse treatment, we had the equivalent of 10 tons of dry mass per hectare being produced by the row plantings. So that was first, actual biomass production, and please note that the highest production was in the most diverse planting. It didn't happen in the least diverse. So we sort of, the idea is to get away from monocrops. Second one was obviously assessing the diversity and the growth of the plants, which we did mainly photographically. Again, taking set photographs every month throughout the trial of what was growing, how tall it was, etc., etc. We developed a technique of measuring the cover crop height using a board, taking a photograph of it and using image analysis to determine how high it was and what percentage cover one had. That sort of deals, and we did a vegetation diversity index. We're doing more vegetation diversity counts. They're coming up. That sort of handles the plants. Then in terms of soils, we carried out Solvita tests. They're very comprehensive. Those included factors such as aggregate stability, Hanley tests to determine biological activity in those soils, and then a host of data on available nutrients. It included soil carbon. So that's the soil out of the way, and then the exciting bits were the biology. So up this row behind me, we had 5 monitoring points. Annually, we've taken soil samples to 300 mils. We take those samples back, they go to NemLab, and they do firstly a nematode count, including plant parasitic nematodes and all the other nematodes. They don't only eat plants. And from that, they calculate a nematode diversity index that is proving to be a very stable measure of how balanced your system is. And we've got fantastic results. After cultivation, it was very unbalanced, and we've now got a 4-year record here and a 5-year record from the Warmbokkenveld that shows that they stabilize over time. We more recently have started doing a similar test with Sporotech on campus at university that measure the bacterial and fungal diversity as well. And then we have more recently also looked at bacterial enzymes in soils.